endoplasmic RAN GTPase activating protein and RAN GEF guanine exchange factor RCC1. It is nuclear guanine nucleotide exchange factor, right? Now, how do we calculate? This is very rarely asked in, but still, how is the transport rate calculated? So, rate is directly proportional to the substrate receptor and one second. Yes, it is your lysine and arginine, RNK. Okay, yeah, it's working. So the transport rate is directly proportional to the substrate concentration multiplied by the receptor divided by the Km plus the substrate, right? Now the next concept is about the organelles. Right, in the cell there are a number of different organelles which help out in the various different functions, right? First is the endoplasmic reticulum. So we have smooth and rough. Rough endoplasmic reticulum is ribosome studded. Protein synthesis occurs over here. Smooth is the one in which we have the lipid synthesis, steroid hormone production, and calcium 2 plus storage. Then we have endoplasmic reticulum stress response. So over here you have the unfolded protein response activation happening in. Then signal recognition particle. So it targets out the ribosomes to the endoplasmic reticulum. For every, let's say, a new particular protein is being synthesized, right, in the ribosome. Now where will this protein go? This depends upon what particular what do we call, what signal is given to it, or we can say what address is getting imparted to it. For example, if it is attached with mannose 6-phosphate, it is going to go towards the lysosome. If it needs to go towards let's say nucleus, so lysine and arginine will be attached to it. If it needs to stay in the endoplasmic reticulum only, then KDEL will be attached to it, so on and so forth, right? Then if it needs to be given out in the body, like if it needs to be like transported out, so it will be then packed into a secretory vesicle via which it will be then exported out via exocytosis. So depending on whatever the requirement of the cell is, depending upon where the that specific protein is required, it will be exported accordingly. And this is one of the segment that you study in the protein sorting part, right? How is the protein sorting done in? Then we have the next segment of Golgi apparatus, right? So Golgi, if I talk about the structure of the Golgi apparatus, so you have the cis and the trans cisternase being there. Then over here the protein modification that is done, which includes glycosylation, phosphorylation, sulfation, depending upon what that particular protein need to work, or what is the role of that particular protein. You have similar modification done in so that it can perform its functionality. Then vesicular transport. So we have COP1, COP2, COP1, coat protein 1, coat protein 2. Then you have clathrine. Then you have caveolase. All of these are helping in the vesicular transportation. There are two types of movement that occurs. One is called as retrograde transport and the other is known as, one is called as the retrograde transport and the other is called as enterograde transport, right? Retrograde and enterograde, what is the meaning of both of these things? So when something need to be transported from the ER to the Golgi, over here there is normal scenario happening in. Over here you have COP1 being utilized, right? Or from ER to Golgi. No, over here you require COP2, and this is your enterograde transportation, right? On the other hand, from the Golgi towards the ER, you have COP1 requirement, and this is called as retrograde transportation happening in, right? So when any of the segment, any of the protein need to be transported from the Golgi body towards the ER, which is reverse scenario, so retrograde transportation, and COP1 will be helping out. On the other hand, from the ER towards the Golgi, the normal scenario, so COP2 and enterograde transportation will be there. Then what are the various enzymes? This is also a part of protein sorting itself, in which you learn about the enterograde, retrograde, COP1, COP2, all of these things, right? Then we have which all enzymes are here. So we have mannosidases, glycosyl transferases, sulfotransferases, all of these enzyme participating in. Then we have mitochondria. So mitochondrial structure, you have an outer membrane, you have inner membrane, you have intermembrane space being there, and then you have the matrix region. What is the it is the mitochondria, which is you must have like learned in class 6th of like 6th NCERT, that it is called as the powerhouse of the cell, right? What do we call it? It is called the powerhouse of the cell, because it helps, like, because now you are in your masters and everything, or you must have uh, completed your masters, you know, ATP synthesis occur, and this is with the help of ATP synthase, which get activated as a result of the proton gradient being formed via the electron transport chain, right? So all of these things must be very much like clear in your mind. So what is the ATP synthesis formula? ADP plus Pi plus hydrogen atoms. It gives you ATP plus water, and approximate this much energy is being formed in, right? Electron transport chain. So you have uh, yesterday also, we like in the previous lecture of one shot revision of biochemistry as well, we revised the entire ETC, the electron transport chain and the inhibitors. So you know that there are 4 different complexes that participates in. Then you have P upon Q ratio, P upon O ratio. So it is approximately 2.5 for NADH and 1.5 for FADH2. Then import into the receptor into the mitochondria is with the help of Tom and Tim. These are the 2 proteins, protein complexes associated. So TOM, TIM. OM, so O is for outer and TIM is for the inner one. Then you have lysosome. So lysosomal enzymes, approximately 60 acid hydrolases are therein, and the optimum pH is around 4.5, which is towards the acidic side. Mannose 6 phosphate is the main targeting signal, which I told you over here. Mannose 6 phosphate. Then autophagosomes. So these are double membrane vesicles that fuse with the lysosomes, and autosomal lysosomal storage diseases. So if there occurs any deficiency, it causes the substrate being accumulated in, right? Then we have peroxisomes. So peroxisomes main function is to make sure that the H2O2 metabolism is getting done, and beta oxidation of fatty acid. In H2O, in peroxisomes, the H2O2 hydrogen peroxide get uh, what do we say, degraded into water and oxygen with the help of an enzyme availability, which is called as the catalase enzyme, right? So catalase reaction is 2H2O2 give 2H2O plus O2. So if you balance it out, it would be the same. Then PTS 1 signal, peroxisomal targeting signal 1. So it is at the C terminal with the SKL sequence for the peroxisomal targeting, and the peroxisome biogenesis. So PEX protein, it mediates the protein import, right? We have various organelles, their key function, what pH do they have, and what are the major marker. This can be a potential part B question, or in any part C question, you might get this information being utilized as a part of the solution for the same, right? Let's continue with the next segment. Now we have the plasma membrane. So plasma membrane is made up of what? First of all, what is plasma membrane? So if I look into the structure of a cell, the outer membrane is your plasma membrane, or we can call it the cell membrane, right? What is it made up of? It is made up of your phospholipids, right? In addition to phospholipids, you have obviously other segments as well, but majorly it is the phospholipids. So this was like explained with the help of the fluid mosaic model, that is the phospholipid bilayer with embedded proteins in it. What is the composition over here? So it is made up of a number of different phospholipids, which includes phosphatidylcholine, phosphatidylserine, phosphatidylinositol phosphate, then phosphatidylethanolamine, right? Four major of these are there, with their concentration differing on the inner and the outer membrane. Then cholesterol is also there, sphingolipid is also there, etc. Then we have membrane asymmetry being there. So different protein lipid composition will be there in the inner and the outer leaflet. And as a result, or because of this asymmetry, you have the uh, asymmetry in the segment and dynamicity in the entire phospholipids, right? Then you have the lipid raft. So this is the cholesterol, sphingolipid, rich membrane microdomains being there. Then if I talk about the transport mechanism, so it can occur via your passive diffusion, facilitated diffusion, active transportation, based upon which segment need to be transported and what requirement is there. Then we have ion channels and the transporters. So passive diffusion, it works based upon the gradient from a high potential towards the low potential. Then you have direction being the downward gradient, high to low, and we have examples as well. Then we have facilitated transportation. Again, example is glucose transporters, primary active ATP, secondary active is the ion gradient. So primary active is against the gradient, that is from the area of lower concentration towards the area of higher concentration. Then we have active transportation, so it is sodium potassium, sodium glucose co-transporters being there. Then we have, like, you have a number of different other segments as well. I'm just trying to touch upon the various pointers so that you can quickly recall all those things. Then we have the microbial physiology. So this is, in this you have to learn about your bacterial cell structure, which includes peptidoglycans. These are cross-linked polymers providing the cell wall strength. Then gram staining. So gram positive, gram negative, two different type of staining are there. Gram positive is thick peptidoglycans, gram negative is thin peptidoglycan plus the outer membrane. Bacterial flagellas, so protein filaments for motility, they are driven by the proton motive forces. Then you have pili also being there. These are hair-like appendages for your adhesion and the conjugation of the two different bacteria for the functionalities, right? Then we have the bacterial growth. So we have different phases of bacterial growth. Starts with a lag phase, then you have the log phase or the exponential phase, then you have the stationary phase, and then you have the death phase being there. What is the generation time formula? So you might get a question from here. Generation time formula is T upon N. T refers to the time and N refers to the number of generations that have been there. Growth constant is this. Exponential growth is calculated with the help of Nt is equal to N naught into T to the power T by G. Then bacterial metabolism. In this you learn about aerobic respiration, anaerobic respiration, fermentation, chemolithotrophic growth, right? Aerobic respiration is when there occurs complete oxidation of the glucose, approximate, not 38, but 32 to 36 ATPs are being produced in. Anaerobic respiration, this is an alternative electron acceptor are being used here. Fermentation is incomplete oxidation. You learn about the various types of fermentation, the various mechanisms of fermentation in unit number 12, applied biology. Okay. Then chemolithotrophic growth, so energy from the inorganic compound oxidation, right? Then you have microbial genetics. So in this we have transformation, transduction, conjugation, plasmids, etc. Transformation is uptake of the free DNA from environment. Transduction is gene transfer via bacteriophages. Conjugation is direct cell to cell DNA transfer via pillars, and plasmids is extra chromosomal DNA elements. They often carry the antibiotic resistance. Then antimicrobial mechanisms. So cell wall synthesis, target is peptidoglycan, example is penicillin and your vancomycin. Then you have protein synthesis, target is ribosomes and streptomycin, chloramphenicol is the example. DNA replication, target is gyrase and quinolones are the example. Then membrane integrity maintenance, targeting is cell membrane and polymyxins are the example. Then you have minimum inhibitory concentration is, or MIC means the lowest concentration that prevents the visible growth. Then you have bacteriostatics. It inhibits the growth, reversible effect, and bactericidal means it kills the bacteria and it will cause like irreversible effect. Then we have something called as the quorum sensing. So it is autoinducers. So various signaling molecules like AHL, AI2, they are all for your cell density communication. Biofilm formation, structured microbial communities in the extracellular matrix. Virulence factor regulation, so many pathogenicity genes under the quorum control. Then environmental stress response, so heat shock protein, chaperones protein induction at the elevated levels, and SOS refers to the DNA repair system that is activated by the DNA damage, REC-A protein mediated, right? So these are the various segments. Now we have few of the formulas also, right? The CDK activity, you have already, like, you learned all of these things in the entire segment also. This is just a quick glance for you all. Then table format, let's quickly recall it with the help of the tables also, right? So till here we had the main segment. You can take this as part 2 also, right? The table format revision of the same. Now we have the various phases G1S, G2M and G0. G1 duration is approximately 30 to 40%. S is also 30 to 40, G2 is 10 to 20. Smallest phase is the 5 to 10% part, which is the M phase. In the G1 phase, we majorly have cell growth, organelle replication, protein synthesis occurring in S phase. Majorly we have the DNA replication, histone synthesis, centrosome duplication happening in G2 phase. Protein synthesis, tubulin accumulation, microtubule organization and all these things. G1 to S checkpoint is the growth factor dependency. Products is the increased cell mass. In the S phase you have the replicated chromosomes being there. In the G2 phase you have the spindle protein being ready and the DNA completeness, that whether the DNA has completely replicated or not, that is checked. In the M phase, you have the spindle assembly checkpoint, 2 of the daughter cell being produced. And G0 is the resting stage in which there is no checkpoint, nothing happening in. The cell is going to exit from the cycle. Quiescence, differential potential will be there, right? Then checkpoints, we have already revised it. So G1 to S checkpoint, then you have intra S, then you have G2 to M, and then you have the spindle assembly checkpoint, that is metaphase to anaphase checkpoint. In the G1 to S checkpoint, the DNA damage, nutrients, growth factors are sensed. Key regulators are P53, retinoblastoma, CDK inhibitors, etc. And what is the outcome? The cell will either be getting arrested or it will undergo apoptosis based upon the outcome. That is, if the cell has the repair possible, so the arrest would be there, repair would be done, and the cell would jump back. If not, the cell will undergo the programmed cell death or apoptosis. Then you have intra S checkpoint. So this is your S phase. In this, the replication fork gets stalled. P53, ATM, ATR, depending upon whether there is problem in what do we say, single strand or a double strand of the DNA, ATM or ATR pathway will be getting activated, and then the replication is halted thereafter if the outcome is not getting fulfilled, right? Then G2 to M transition. So this is G2 to M bound. At this occurs when the cell is entering from the G2 to M part, right? So from G2 to M over here it would occur. So DNA damage is checked, incomplete replication is checked. These are the various regulators, and the entry is blocked if the response or whatever the requirement was there gets remains unfulfilled. Then we have the spindle assembly checkpoint. So location is metaphase, kinetochore microtubule attachment. So MAD2, BUBR1 securin, and over here the anaphase gets prevented, right? Then you have various cyclins, various CDKs participating in. So you have various cyclins, various CDKs. They make up a complex together, function in, and we have the various inhibitors as well. P21 you can see is an inhibitor in each one of these because that's very important. Then mitotic stages in detail. Prophase, sometimes in some books you'll get to see pro-metaphase also, and then the main metaphase, then anaphase, anaphase A, anaphase B, telophase, and then the cytokinesis. Two things are there. One is called as karyokinesis and one is called as cytokinesis. Karyokinesis means when my nuclear content is getting divided, right? The entire division that is happening, that is your karyokinesis happening. And your nuclear content is getting doubled and divided into half thereafter. Cytokinesis is when your cytoplasm is getting finally distributed into two and two cells being formed in, right? So prophase is condensing. Nuclear envelope breaks down, spindle status assembling. Duration is approximately 10 minutes, and these are the features. In the similar pattern, you have the various features, their durations, spindle status, what will happen to the nuclear envelope, and what is the chromosome state over here you can read about. Then the difference between the mitosis and meiosis. So in the mitosis, you have one division. In meiosis, you have two divisions being there. Chromosome number is deployed, like the chromosome number will remain the same. In meiosis, it is going to get half. Homologue pairing. In mitosis, no homologue pairing. In meiosis, yes. Crossing over, not there in mitosis. In meiosis, it is there, which is because of genetic recombination. So, variation, the exact copy number, clones are being produced. In meiosis, there is no genetic, the genetic recombination is there. Then daughter cell number 2 and over here 4, cell function. So growth, repair, and asexual reproduction, and over here the main sexual reproduction being there. Then you have recombination, no recombination, and over here it occurs in the prophase 1, and in prophase 1 also exclusively in the Pachytene stage. Yeah, then you have microbial structure dynamics portion, the various segments. Then you have actin microtubule filament, the binding protein, very important. Take a screenshot of this or just keep a note of this. The various actin binding proteins, so proflavin, coflavin, myosin, thymosin, beta 4, ARP23, formin, capping protein, vilin. You can easily get a part B or a part C question from this segment in any manner. In the recent papers, we have got questions something like you're given some introductory information, few names of the proteins given here or some additional one, and they can ask you which of the protein have a direct role in the actin, which of the protein have a direct role in the cytoskeleton movement or nuclear transportation or any of the format. So all the actin binding proteins, intermediary filament, the type, tissue, where do you find them, diameter, molecular weight, function, disease association, all these are very important. See, even this particular slide is very important, where you have the nuclear pore complex composition. So how many different nucleoporins are there, how many different NUPs are therein, what role do they have, like what function do they have, how many types, and what's the molecular weight of each, can easily be asked in. You can easily get some experimental question also designed in from this segment, so make sure that you remember it. Then how does the nuclear transportation occur? So we have the CRM1 export, we have carrier mediated import, we have classical import in RAN export, SNRNP import, etc. happening in. Then you have endoplasmic reticulum structure and function. So smooth, uh, rough, and transitional is also there. Then various endoplasmic reticulum protein synthesis and targeting. So the signal peptides, inter internal stop transfer sequence, C-terminal KDEL and lysine based sorting. So what is the location of these signal sequences? What do they recognize? What is the direction of each one of these? Is translocon helping in which coat is there, and what is the final uh, target of the same? All these are very, very important, right? Then you have Golgi apparatus, organization and function. So cis Golgi, medial Golgi, medial or medial, whatever you want to pronounce it. Then trans Golgi, and then the trans Golgi network is also there. Which all different enzymes, very important. What modification do they cause? What is the pH, and what is the final output? Then vesicular transport proteins, mitochondrial structure complex. I've tried to add as many things. See, n number of tables are here, and all these tables, I'm telling you, you can easily get a question from this particular portion. I'll provide you with this entire table for your revision for your segment, right? I'll upload it on the Lakshya batch as well. You have it in the batch, like in your own batch, obviously. So, and just let me know if you want me to upload it on some other area also for easy access. I'll do that, right? So that's about this entire revision segment. If you have any doubt, any problem in any of the segment, you can ask in. Otherwise, this was one shot entire revision. I'm not reading out these all parts. You can quickly go through this. You'll get this PDF, and you can easily access it for your theoretical revision portion, right?